Thank you very much, Rosie. Can you hear me at the back? Great. My voice is not a certainty in all this because I've got a cold on, so we'll see how this goes. What I want to talk about today is a certain kind of project which, within the broad church of ecology, involves the following of individuals over entire lifetimes and many individuals over many generations. And what I want to do, by example from my own projects, is to show how I think these projects can contribute to our understanding of how the world works. Crucially, while I do understand that experimental analyses and common garden analyses and many other fine approaches to ecology exist, I think studying organisms that are actually in the wild and accepting all the insults that the weather and density effects throw at them has its role in this world as well. So it's conventional when introducing this kind of study to think about the very long-term studies of small passerines, particularly great tits, in, for example, the Wytham Woods and the place I can't pronounce in the Netherlands, as the sort of type specimens for long-termism. So it amuses me, as a great fan of New Zealand, to introduce you to this chap, who is called L.E. Richdale, who very substantially predates some of our European studies. So between 1936 and 1954, he studied over 200 pairs of a kind of a penguin that lives in the Otago Peninsula in New Zealand and wrote up this monograph. There's a really nice recent biography of him. And I mean, I think if it wasn't for the fact that this project had ended, we'd be thinking of him as the hero of long-term studies of individuals. But if you read the biography, what's even more fun is it turns out he got his inspiration from a woman. So Mrs. Margaret Morse Nice, what a great name, had been studying the song sparrows using colour-ringed individuals in her back garden before L.E. Richdale got going, and meanwhile raised 5 children. And I just love this hat. I think it's a marvellous way to be doing real research. Okay, so what I'm going to do today is sort of 3 topics, really. I'm going to introduce my field studies. Then I'm going to go through a series of questions that we might be interested in asking in ecology and evolution, which, to my mind, represent successive dives down into more and more detail about the individuals and their genetics. Then I want to make a particular point about our need, I think, now to study and understand temporal trends. And then that'll be the end of the talk by the time we get there. So introducing my study populations, we are off the bottom right of this map at the moment. And I have 2 study populations, one on this diamond-shaped island called Rum, and there we study deer. We study them in the North Block that's labelled 4 there. And also out in the North Atlantic, in the archipelago of St Kilda, we study the sheep living on the main island Herta in a particular area called Village Bay. And these are these little primitive domestic sheep which have lived wild for millennia on these islands. I want to say this is kind of an antithesis to what Luigi Boitani was talking about yesterday. These are predator-free islands. What we're doing here is studying herbivores in a simple ecosystem where their main interacting organisms are the plants and the parasites, and there are no predators to, as it were, create another trophic level, and we're very much studying a simplified ecosystem. Of course, the key thing in these sorts of studies is utter routine in collecting data. So the routine that we employ on these 2 studies is absolutely dogged, and very, very good field workers are involved. So on Rum, we really bust a gut every year to label up all the calves as Christmas trees early on in their lives. We then follow them through their lives by censusing them to see who they are with, where they are, and so forth. We observe the rut in enormous detail to see which stags have got which hinds in their harem. And in the winter months, we monitor for mortality and also, in the case of the deer, search for cast antlers, which is literally like finding needles in a haystack. But near to here is a collection of many hundreds of cast antlers from the rum study. On St Kilda, we have very much the same routine, intensive effort to mark all the little lambs when they're born. The difference is that we're able to round up the sheep once a year, or at least a substantial proportion of them, and do a great deal of phenotyping and sampling on them. And then, of course, we do observe the rut, again, in great detail. And we can also find mortality. And I suppose a unique feature of these studies is that because these are landlocked mammals that can't fly, we do actually find a very high proportion of the individuals that die. So thinking about the kind of history of these projects and how they've developed over the years, the first thing to say is I think they've contributed a great deal to our understanding of what I'm calling here basic ecology. So what are the effects of weather and density on individuals and populations? And how do these effects build into population dynamics? So I think it's fair to say that if you want weather effects, we can show you weather effects. Now, I'm going to dodge mercilessly between deer and sheep in this talk, but I'm always going to try and have a picture of what I'm talking about somewhere on the slide so you know whether I'm in deer or sheep. This is a fairly old slide showing effects of early autumn rainfall on mortality of calves and yearlings on rum, and each dot is a year, and demonstrating very clearly that the weather has an effect on the lives of deer. This is but one effect that I could have mentioned. You want density effects, we can show you density effects. This is a similar kind of a plot showing mortality to 2 years of age in relation to the number of females using the study area, which turns out to be one of our best metrics of density. And you can see here a very clear relationship between density and mortality, and you can also see the quite well-known result that in these sorts of organisms where males are bigger than females for reasons of sexual selection, males are actually more vulnerable. Now, the vagaries of density and weather in different years, of course, causes variation in the recruitment rate from year to year. And if you have individuals, you can now look at the way in which cohorts vary from year to year, and you can see the effects of recruitment of large versus small cohorts. So in this glorious Technicolor picture from Tim Coulson, you can see the effect, for example, of the recruitment of a very large cohort of yearlings in 1986, which really sent the population up. So you can get at the underlying structure of the population and how that's feeding into the population dynamics. And I'm now going to switch to the Soay sheep because they have much the more interesting and dramatic population dynamics and show you how these weather density and population structure things build together. So this is the time series of individuals living in our village-based study area on St Kilda from the start of our project in 1985 to about 2000. And by this time, we knew, and also from previous research, that there were very substantial crashes in the population. And thanks to the good offices of Brian Grenfell particularly, we now knew that this was due to overcompensating density dependence. In other words, the population could grow so high in a single breeding season that it would greatly exceed the carrying capacity of the ground in the winter, and that's when you would get a population crash. But I think a very interesting and illustrative analysis was then conducted by Tim Coulson, who showed us how exactly the weather and the density and the population structure, the demographic structure, interacted to generate the population dynamics. So in this plot here, we've got 3 things varying. We've got, first of all, a population size scale on this axis. so low, intermediate, and high. And on this axis, we've got an index of weather. Actually, it's the North Atlantic Oscillation, which generates winters of different horribleness and niceness. And on each of these little plots, there are 4 lines representing something about the demographic structure of the population. And they, in this case, represent what proportion of the population consisted of prime-aged females, which are the most resistant to death. And I like this plot a lot, because what it shows is what the population typically does in any of these 9 circumstances depicted. So if it's a good weather winter and the population density is low, they always go up. If it's a high density winter and a bad winter on weather, then they go down. But there are areas within this plot, and particularly this middle one, where you can quite clearly see that what happens varies according to the demographic structure of the population. So I think this was a sort of key insight that we were able to contribute to this basic ecology set of questions. Quick sip of water. Okay, so my next set of questions, getting a little bit more detailed about the lives of individuals, is to think about life history questions. So for these kinds of questions, I think the key thing that we can provide on these sorts of projects is events at point A in the individual's life, followed by knowledge of what happens at point B, and in some cases C, D, and E. So this is where you know repeated events or consecutive events in the lives of individuals. So, for example, key questions here in life history questions would be, what are the costs of reproduction? You know, what are the consequences of reproducing in a particular year for the future of that individual? How plastic are individuals? So this is whether you can detect that individuals vary in their response to things over their lives. Do individuals senesce? This is a very popular study topic at the moment. And so these sorts of projects, including ours, have contributed, I think, rather a lot to this kind of field. And I can show you here a cost of reproduction type result. So once again, we have on this axis something about the late summer rainfall, which we know is a bad thing if there's a lot of it. And here we have the probability of survival for a mother dependent on the situation with her previous calving. If she did not calve at all, or she lost the calf when it was very young, then her survival is fine, whatever the weather. But if she had a calf and whether or not it survived or not through the winter, if she reared it into the winter, then her own survival is compromised under these conditions of bad weather. I should point out to any PhD students present that I really strongly doubt that any editor of a BES journal would let you put this plot into a paper these days. I think we would very much wish to see some error bars and things around this. My next topic then was to think about plasticity, and I cannot overemphasize how important I think plasticity of individuals in response to what the environment throws at them is absolutely key in some of the topics I'm going to come up with next. So on rum with the deer, we have a classic plastic trait, which is that spring temperature is associated with birth weights. This makes perfect sense from the biology of the animals because it's known in mammals that the growth of fetuses is greatest in the last trimester of pregnancy. And this result has been discovered many times in different analyses of the data from the project. And these points here are annual means. The key thing here is that we can now investigate using individual data whether the overall population effect, what you see from the black data points, is also true at the individual level, or whether individuals have different kinds of plasticity. Are some more reactive than others? As it happens, in this particular case and this particular analysis, it would appear that because the within and between slopes are very similar, then we don't see any particular variation for this plastic trait in the red deer. But in other studies of other organisms using the same sort of approaches, people do find individual variation, and you can then ask, well, what is it that is causing that individual variation? Does it have to do with how good a start they had in life? You can also, to jump forward a bit in my talk a bit, think about whether the plasticity has an inherited component. And finally, in this section, we have a great deal of research going on at the moment in these kinds of projects having to do with senescence. So here's a few plots from a recent paper. I'm particularly pleased to have published something in a journal called Experimental Gerontology, because there is absolutely nothing experimental about this data. This is observational data. So Adam Hayward analyzed here, in fact, 20 different traits in the Soay sheep. What the plots that you're looking at are how the trait changed in the sort of second half of life. And what you see here is something that I think is now well established, which is that wild animals show senescence. That is to say, in an example like this, you can see a decline in the trait long before the animal actually dies. One of the points of this particular paper is to show that actually different traits decline at different rates. For example, if you compare birth weight here and parturition date here, it would appear that birth weight is declining quite a long time before an animal dies, whereas parturition date appears to sort of crash in the very old. Now, if you've got longitudinal data on individuals, you can investigate these sort of very general patterns in much greater detail. You can, for example, distinguish whether these are genuine effects or simply the effects of selective death of individuals with particular traits. You can, for example, try and understand why it is that different individuals age at different rates. And there's a lot of developing literature from long-term studies which demonstrates, as an example, that reproductive effort early in life has a strong effect on the rate of decline later in life. That's just an example of some of the sorts of effects that have been detected. Another thing you can do with this kind of data is ask whether it is really actual calendar age that matters to the animal, or how soon before it dies that is causing these sorts of results, which you can't do with cross-sectional data. You simply have to have longitudinal data. So those are the sort of topics I feel you can address with this kind of longitudinal individual-based data in connection with life histories. But now I want to dive down another level and address an area which has really come to fruition in the last few years as a result of a maturing of these long-term projects and our ability to do some genetic analysis on them. So in particular, I want to talk a little bit now about the area of evolutionary quantitative genetics. So this is an effort to understand, using these data, stuff about the genetic architecture of the traits we're looking at. So I'm particularly interested here, and I mean here, things like what is the heritability of a trait? What about the genetic correlations between traits? What about inbreeding depression? I'll come back to these topics in a moment. Then questions about whether we can measure selection satisfactorily, whether we can predict, or do we predict a genetic response to selection? Can we even see a genetic response? Can we detect a genetic response to selection? All operating within the long-term studies of individuals that I'm talking about. Now, to remind anyone who doesn't recall, the heritability of a trait is the proportion of phenotypic variance of that trait, which is due to additive genetic variation. The key thing to remember here is that additive genetic variation is the kind of variation that will respond to directional selection. So if there's additive genetic variation for a trait, and then there's selection for a trait, you might predict that the trait would change. And the method for estimating heritability traditionally has been to compare the mean of the parents with respect to the trait with the mean of the offspring with respect to the trait. And as you can see from this picture, this approach has been deployed in, this is a Darwin's finch, from a long time ago in this very long-term study of the Darwin's finches run by the Grants in the Galapagos. And if you see a strong correlation, such as is shown here for 2 different years, what it says is that the offspring's beak size, in this case, is very strongly determined by its parent's beak size, which probably indicates that it has a high genetic control in terms of additive genetic variation. Now, I think we now know that even in passerines like Darwin's finches, it's often the case that the pairs that you see feeding the chicks may not be the true parents. So this is what we call a social pedigree that was used here. In my kind of animals, you absolutely don't have a social pedigree. It's very, very hard to know from observation who is the father. And in consequence, a great deal of my life has been occupied with using essentially each new genetic marker as it came along to try and derive parentage for the animals in my study areas. So finally, I think we're getting pretty good at this. So here is the pedigree of red deer on the Isle of Rum. And what you have down here is each cohort of calves as they were born. And what you have is a blue line connecting a father to its offspring and a red line connecting a mother to its offspring. And this kind of assembled parentage information has provided, I think, a pretty good breakthrough towards investigating this field of quantitative genetics in much greater depth in wild study populations. I just want to make one point about this pedigree, which comes up later. This may look like an impressive pedigree, but apart from, in my opinion, the song sparrows of Mandati Island, all wild pedigrees have error. And in the case of our pedigree, the errors include missing links because we can't catch and sample the stag that we want to. But also, a pedigree is always defined by its top generation. And of course, we don't really know how related individuals at the top of the pedigree are. And nor do we know the relationship between any immigrants who come into our study area from other parts of the island and the rest of the pedigree. So I just want you to hold the thought that lovely as this is, it's not perfect. The other development which came along at the sort of same time as pedigrees for wild populations were being established, especially by genetic methods, is the ability to use really advanced quantitative genetic modeling from the animal breeders to develop more precise estimates of heritability. So I recently showed you a slide looking at a parent-offspring correlation. The so-called animal model, which is a special kind of a mixed model, allows you to use all the information in a pedigree relating to an individual. So you can use not just the parents, but the grandparents, the cousins, the brothers, the sisters, everything. So you derive a heritability with the best possible estimate and lowest possible standard error. And also, incidentally, because I shall come to it, you get for each individual a so-called estimated breeding value for the trait, which is an estimate of the genetic merit of that individual for the trait. And if you were breeding cows, you'd be interested in those estimated breeding values. So let's see how this pans out with a particular trait on the Isle of Rum. And I'm going to talk here about cast antler mass. So we've got all these antlers. We know who they come from. We have the pedigree, and so we can partition the variance in cast antler mass between different sources of variation. And inevitably, a lot of it has to do with age, because stags grow bigger antlers with age. But also, you can see considerable contributions from the environment, so the permanent environment, that's things to do with perhaps the early life experience of the individual, and the year of growth, so in a good year they grow bigger antlers. And crucially, 18.2% is the heritability for antler mass in this particular case. So the heritability in principle can range from 0 to 100%. So that's the scale we're thinking of, and here it's 18.2%, which is, I would have said, about medium for a trait in a natural population. While we're on this pie chart, I think the thing to focus on here is that we actually spend a lot of our time when modeling these traits thinking about the other potential sources of covariance between individuals. So thinking about the year of growth, for example, thinking about the year of birth, for example, and not shown here, but in many other studies, thinking about the consequences of having the same mother, so maternal effects. We really try as best as we can to differentiate the genetic effect away from all the others. Okay, so the reason why we're interested in the heritability of a trait has to do with the fact that we would like to be able to predict whether we're going to see a genetic response. And for this purpose, we started, as a field, I'd say, we started with something called the breeder's equation, which is a nice, simple equation, which simply says that the response to selection is the product of the heritability, which you already know about, and the selection differential, which is a measure of selection. This equation has a multivariate form, which I'm not going to spend time showing you, but crucially, traits are under joint genetic control very often. They are not free to evolve independently of each other. And so really, when you're considering a response to selection, you should think about the heritability of all the traits, the genetic correlation between the traits, and the selection on all the traits. So I would say that the principal, not sure about discovery, but revelation of the field of evolutionary quantitative genetics as applied to wild animal populations has been to demonstrate that actually this equation doesn't work terribly well in natural populations. And that is principally because of various ways in which the ecology gets in the way. Well, I don't have time to speak of many of those ways in which the ecology could get in the way, but I'm going to just go through one as a matter of interest. So if you think about cast antler mass, we might be interested in thinking about, well, what's the selection like on cast antler mass? Because we now know the heritability is about 18%. And if there is also selection on antler mass, we might be predicting that there would be evolutionary change in antler mass. So if you do a plot of the raw data, so this is antler mass from the cast antler data, and this is the lifetime breeding success of the owners of the cast antlers, as measured by our parentage inference methods. And you can see that there is a strong positive correlation. So there would appear to be some selection on antler mass. However, in this particular case, if you then drill down into where this correlation is coming from, you discover something rather interesting. You discover that if you look at the breeding values, so that is the genetic merit of those individual stags for antler mass, and compare it with their breeding success, there's actually no appreciable correlation at all. Whereas if you look at the alternative view, which is the environmental deviation, that is the variation that is not due to the genetics, and the lifetime breeding success, you discover that there is indeed a correlation there. So the take-home message from this is that the selection that we see, and is undoubtedly happening on the phenotype, is not actually targeting the genetic variation for antler size. So it's a classic example, perhaps, of the third variable problem, where you've got something else that is driving the situation. So we speculate that it's probably nutritional, So good nutrition both increases your antler mass and probably improves your lifetime breeding success. And the apparent correlation that you might think had a genetic component does not. An alternative and perhaps more elegant way of showing these kind of effects is to compare different ways of predicting evolutionary change. And I'm now going to switch to a case involving Soay sheep body weight. So we have a rather primitive method for measuring Soay sheep body weight. You take a bathroom scales and you take one Alistair Wilson and you give the sheep to Alistair Wilson and you then weigh him with and without the sheep. And that's how you get the weight of a sheep. So Michael Morrissey did a very nice analysis where he compared the predictions that you would get for Soay sheep body weight using either the univariate breeder's equation, which I showed you earlier, which would predict a lot of change, a response to selection, the multivariate breeder's equation, thinking about various other traits that might be tied in with body weight, which gave you a similar sort of prediction, and the one measure which would tell you whether the selection is targeting the fitness, which is the genetic correlation between body weight and fitness. And that measure predicts no evolution, okay? So that's a really interesting difference between these various predictors, which is almost certainly a issue that has been, as it were, provoked by the ecology of the situation, that the selection is not targeting the genetic variation for this trait. So then you might ask, well, okay, have we actually measured any response to selection in this trait, in this case, body weight in Soay sheep? And in short, the answer is no. So it took much time and thinking, especially by Jared Hadfield, to work out that we must be very careful about trying to estimate genetic trends using estimated breeding values. And here is the neutral evolution distribution of expected change in body weight on St Kilda from an analysis he did. And this sort of grey line showing here is what we actually observe. And there's no way that what we can see is any different than a no change in body weight at the genetic level. It would have to be out here or out here for us to see a genetic change. So I think this is pretty much in common with many other wild studies, that we find it very hard to demonstrate genetic change in traits in natural populations. If you've been interested in these kinds of issues and want to learn more, I do recommend this book, which was recently published, bringing together information on quantitative genetics from many natural populations. Okay, diving down another level then, there is, I think, an interesting set of questions to ask about evolutionary genomics. So I mentioned already that the pedigree is not perfect. Genomic estimators may change the genetic architecture that we've been looking at, the heritabilities, the genetic correlations, the inbreeding, and so on. And genomics also allows you to identify loci. In principle, it allows you to identify loci underpinning the trait, so diving down further into the architecture. So I have embarked on this kind of exercise, and I'm very lucky because there's such a thing called the International Sheep Genomics Consortium, and I think this is absolute magic. This object here is the size of a microscope slide, and each of these little channels here will analyze the DNA of one sheep at 50,000 places, evenly spaced throughout the genome. This is an absolutely overwhelming amount of genetic information about individuals. And little do the patients of the Western General Hospital in Edinburgh near here know that several thousand Soay sheep and deer have gone through the human genotyping lab there. So I mentioned that the pedigrees are not absolutely perfect, and here is what I mean. This is a relationship between our best pedigree relatedness between all individuals, and a genomic relationship here. And here you can see, for example, in this wodge here, some individuals for whom we've got the wrong father in our pedigree. And over here you can see individuals which we thought were unrelated in the pedigree, but actually it turns out they are related to individuals in the pedigree. The genomics is telling us that they are related to various individuals in the pedigree. So there's no question that a pedigree is not quite perfect in populations such as ours. So the question is, does this make a difference to any of our estimates of heritability, etc.? To which the short answer for most traits that we've studied is no. So this is the heritability of adult weight, and these are two different pedigree estimates and a genomic estimate, and the answer is no. It has made a difference in some rare cases, particular traits, particularly with regard to maternal effects, but not much. We've had much more excitement, I think, with thinking about inbreeding. Now, I haven't spoken about inbreeding very much so far, but in measuring inbreeding in a pedigree, you absolutely have to have an unbroken chain of relationship between two individuals who are the parents of your inbred individual and the common ancestor. And in our kind of pedigree, that chain is often broken. And so this is a relationship here between a pedigree inbreeding coefficient and a genomic inbreeding coefficient, showing that once again there is a lot of noise in this. And in particular, if you look at this distribution up here, with genomic inbreeding, with pedigree inbreeding, you have an enormous number of individuals with zero inbreeding. So that's because the chain is broken somewhere. Whereas in a genomic estimator, you get basically everybody on a continuous scale going back to ancient history in terms of the pedigree. And you'll also notice it's got a much, much nicer distribution for analytical purposes. And these inbreeding metrics have had a big effect on our ability to measure inbreeding depression. So here you can see inbreeding depression in juvenile survival in red deer. And whereas we knew there was inbreeding depression in a calf, we didn't know that the mother's inbreeding coefficient has a profound effect on its survival, which we now know. And we've also been able to measure quite severe inbreeding depression over the lifetime in both sexes, which is by no means as clear from the pedigree approach. Diving down yet another level, I mentioned an interest in looking at individual underpinning loci for traits. And I'm going to move rather swiftly to one trait with which we've had a lot of fun, which is horns. So horns are clearly offensive weapons, particularly in males. And yet, shockingly, on St Kilda, in both males and females, you find individuals without horns. So this guy is a mature ram with no horns, next to a mature ram with horns. And our key question here was, why on earth should this variation exist? It was clear this was a relatively simple polymorphism. It's a really profoundly different trait in different individuals. And as a result of our genomic information, it was possible for Susie Johnston to show, using a genome-wide association analysis, that this trait was controlled on chromosome 10 of the sheep. So this is a test of every single of the 38,000 polymorphic markers as to where there was an association with horn type. And much work later, so I have to move quickly on, Susie was able to demonstrate that this was under the control of a single locus, and that normal horned rams come in two sorts. There's the guys with the really long horns, who are what we're calling plus plus genotype. There's guys with intermediate length horns who are plus P genotype. And then if you have the PP polled gene as a homozygote, you either grow rather stubby little normal horns, or you grow wonky little horns or no horns at all. Knowing the genotypes of individuals is absolutely crucial in understanding selection on loci and traits like this. So what Susie was able to show was that there was a very interesting set of relationships going on here. So if you look at adult reproductive success, really not surprisingly, the guys with normal horns, these two categories here, have much higher reproductive success than the guys with the little horns. The guys with the little horns are not able to get as many matings. However, if you look at survival, the guys that are the plus genotype have lower survival than the other two genotypes. And as it turns out, if you put these two together into overall fitness, the guys that are the heterozygotes have the highest overall fitness. And this is therefore a rare example of heterozygous advantage in a natural population. Really instructive to know the underlying loci. It is, of course, much, much more of a challenge to look at quantitative traits, so truly continuously varying traits, and we have had a go at this. For example, we do have a very large quantitative trait locus, giving us a huge difference between the two homozygotes in terms of leg length. This is a larger difference here than between the sexes on St Kilda. But alas, the allele frequencies are such that we can't really detect selection on this locus, and nor is there much going on in terms of its allele frequency. But I think it's instructive to see that you can get down to this kind of information. So I want to end on a short section, really, on temporal trends. It seems to me that analysis of natural populations is now revealing masses and masses of temporal trends. The existence of trends in phenology don't need much comment because they're everywhere at the moment. I'm going to show you some other kinds of trends in terms of other traits. And of course, there's a lot of interest in phenology mismatches and population sizes. And I think that one of the things that these long-term studies are in a position to do is to try and understand the relationships between these various trends, and also try to have a go at the mechanisms that underlie what we observe. Part of my getting into this topic a little bit is just to pass comment on a field which is now known as eco-evolutionary dynamics. These are all good friends of mine, and this could have been one of many, many papers. But I am, as an evolutionary biologist, quite bothered by statements that we now think that ecological and evolutionary changes can occur on the same time scale. I think we need to think much more carefully about some of the relationships here and whether this is really a realistic thing to say. And I think this is partly motivated by the fact that, for me, an evolutionary change requires there to be a genetic change. In fact, temporal changes, it seems to me, can occur for 4 reasons. You can have immigration, or indeed emigration, of different phenotypes and genotypes. You can have demographic effects, like suddenly a cohort is particularly large and has a big effect on the population. There's plasticity, which we've looked about, and there's microevolution. And as I've shown you, this is a very hard thing to show. And I think, as an evolutionary biologist, I'd like us to keep microevolution for a specific genetic change in the trait. So we've seen a number of trends in our data sets. Famously, for example, we've got quite large phenological change happening in our study population of deer. I would particularly draw your attention to this one here, which is birth date and indeed estrus dates. The calving date has got 12 days earlier in the time frame you're looking at there. And yet, as a mystery in this particular paper, we couldn't see any obvious change in the survival of calves. And so in a more recent paper, Katie Stouffer conducted an incredibly detailed and careful path analysis for red deer birth traits, and you're in no sense meant to get into the detail of this. But the conclusion of this careful path analysis is that survival via the indirect effects, particularly of birth date and birth weight, would be predicted to go up, but was exactly counteracted by direct effects of, in particular, a small increase in the population size over time. And so the reason that we're not seeing any change in the trait of survival, juvenile survival, has to do with exactly counteracting effects between different parts of the model. Another major trend we're observing in the Soay sheep has to do with the fact that the mean weight, or indeed hind leg length of individuals, is getting smaller over time. This is absolutely astonishing. This has been coupled with what appears to be a rather large increase in the population size on the island, both in our study area and on the whole island. And various people have so far had a go at trying to investigate what's happening here. Arpat Ozgul led a paper which tried to decompose the mean weight of the population in any one year into different components. So the major component was, of course, the structure of the population in terms of sexes and ages, but a second component was about growth, this one here, contributing 18.5% to the change over time. And this was because the lambs were not growing as much between particularly first year and second year in the population. And even more recently, Tim Coulson has been applying these integral projection models to the population, thinking about the 4 transitions that you need in a very, very detailed matrix model to predict forward from the current population to the next year or whatever, a survival function, an offspring number function, a growth function, and this one which causes evolutionary biologists a bit of trouble, which is called the inheritance function. And this is a very interesting way of thinking about how the population might change over time. And in a more recent paper here, the authors have looked at how the NAO, the North Atlantic Oscillation, plays into this, and concluded that actually when the NAO is positive, the population will go down, but the mean weight will go up. When it's negative, the population will go up and the mean weight will go down. So it's possible that what we're seeing at the moment on St Kilda is one particular sort of wave of the NAO going through. I think that these are but a couple of efforts at understanding some of these long-term trends, and I think that my projects and others, and of course there are many other projects involving long-term studies of individuals, we should get together and try and understand how these trends are happening, and we should try and work out whether we can get any evidence at all of any genetic change in our populations. So to summarize, I hope I've convinced you that there's a great diversity of questions that can be addressed in these long-term studies of individuals. I've had no time to mention behavior, parasitology, immunology, and indeed management implications of the things we've done. I do think that in future research, as I've just been saying, we need to get at the mechanisms underpinning temporal trends. I think that's one of the big things we can do. And the other thing that is going on quite a bit, you can now do an omic if you study a trait. It's called phenomics. And we are doing, with my colleagues anyway, are doing a great deal of phenomics on archived blood samples and things, which I think is going to be a major area of development in the near future. And if you want to find out more about what we're doing on the Sheepy project at the moment, come back here at 11 o'clock. So finally, I'm just going to put out a slide of all my acknowledgements and my current funders, but I'm not going to go through it. Thank you.